Okay, so the eye has specialized receptors that absorb and convert light into neuronal response. And, they look very unlike, your classical kind of neuron that we've been describing, that's got its dendrites for input, its cell body over here with the nucleus, and the chromosomes, its axon hillock, which is where electrical message gets initiated, an axon, which is the sort of wire-like projection, and then these terminals where the electrical message that goes along the axon is converted to the release of neurotransmitter at the synapse, right? Actually, these cells look very different. So here's one example of what we call a cone cell photoreceptor. This is a photoreceptor that's going to absorb electromagnetic radiation within a very narrow spectrum, because the electromagnetic spectrum is quite broad and the sliver that correlates with visible light is quite small. And we have 3 of these, what are called cone cells. This is a pipe cleaner version. I'm going to get you another one that's made out of plaster here that was created by a student at Portland State. This is like what they call the outer segment of the cone cell. The cones actually have sort of cone-like outer segments. They kind of, transition down to a point here. And what the significance of that is that there are membranes that form these discs in the outer segment that are, and embedded in those membranes are actually proteins. And these are proteins that are, manufactured from the cell body, which is weirdly right over here. So this looks very different than a classical neuron that we were looking at. And these proteins are called photopigments because they will absorb, different wavelengths of light within that narrow range of the electromagnetic spectrum that correlates with visible light. So this is actually what they would, we would call an S-type cone, because it absorbs the short wavelength within the range of visible light, which is at the violet or blue end of the spectrum. And so when the pigment that's back here in this cone-like segment, absorbs, the short wavelengths, within that range of visible light, it converts that energy of electromagnetic radiation into kinetic movement. That the protein itself moves, and that movement results in the release of a chemical, which will then initiate a series of changes inside the cell. It'll go from one chemical and do something to another chemical and to another chemical, and it sort of initiates what we call a biochemical, because these are chemicals associated with life, cascade of changes, right? And the end result is actually bizarrely, and the eye is paradoxical in many ways, it is the shutting off of release, the inhibition of release of neurotransmitter here from the end. So this is what they call the paradoxical dark current. Your photoreceptors are actually the most active. They fire the most, intensely in the dark, in the absence of light. So when you close your eyes at night, when the sun goes down, when you turn off your screens, basically that's when these photoreceptors are firing the most. But paradoxically, that firing is inhibitory. It inhibits, the next cell in the chain, which is the bipolar cell, which links to the ganglion cell. And those ganglion cells, their axons swirl around and leave the eye and form the optic nerve. So there, it turns out, in terms of photoreceptors, there are 3 distinct types of photoreceptor typically. There is the short wavelength photoreceptor, where the photopigment that's expressed will absorb short wavelengths of visible light, which correlates with the violet or the blue end of the spectrum. Then there's the medium wavelength absorptive photopigment. These are called M-type photoreceptors, and the medium wavelengths within the range of visible light correlates typically with our perceptual experience of green, green over here, right? And then there is the L type, or the long wavelength, type of photoreceptor, which absorbs, the long wavelengths within that overall range of visible light spectrum. And that typically correlates with a perceptual experience of red light. Then there is another kind of photoreceptor. I don't have an example here, but instead of having a cone-like kind of, outer segment. It has a rod-like outer segment. It's much longer. It's much more extensive. It doesn't sharpen down to a point. And what that does is, first of all, the photopigment that's expressed in the rod cells is one that will absorb, light within the entire range, electromagnetic radiation within the entire range of visible light. So it's not so selective. And it also has many, many more, photopigment surface area regions, because it's got this rod-like structure as opposed to like a sharp cone-like structure. So it turns out that rod cells are really good at absorbing light when there's lower levels of light, like let's say at dusk or at dawn. And it turns out that rod cells are found primarily in the periphery of the retina. And many, many rod cells are linked up to, a single bipolar cell, and then to a single ganglion cell leaving the eye. So rod cells are particularly good in low light conditions, when something is moving across several rod cells, because that'll be more likely to set off, to remove the inhibition, to stop more of those from firing, and to remove the inhibition off of output from the eye. So rod cells are particularly good at peripheral vision, at night vision, and also at motion detection, basically in the periphery. While the cone cells, those often three types, are actually found primarily in a portion of the retina known as the fovea. That's that divot, right, where the bipolar cells and the ganglion cells are kind of splayed to the sides, and light is more direct entry. And these cone cells are tightly packed in the fovea, and they're linked to small little bipolar cells, and then to smaller ganglion cells too. So even though it's interesting, you've got about 90 million rod cells out here in the periphery largely, and you only have about 4.5 to 6 million, cone cells. The majority of the axons that are leaving the eye in the optic nerve are actually carrying foveal information from the cones. And the fact that you have three cones typically, some people have two, some people have one, some people actually may have a fourth cone photoreceptor, means that you can sort of compare patterns of activity, right, across several labeled lines. The S-type blue, the M-type green, and the L-type red. And so the central foveal vision is critically important for color detection, because you're actually comparing the pattern of activity across these different wavelength specific kinds of photoreceptors, the cones. And also you need daylight. You need more light, because the cone shape means there are fewer pigments that are capable of capturing electromagnetic radiation and generating an actual response. The other thing is that because they're so tightly packed, and it's more of a kind of 1 to 2 to 1 output, in terms of the photoreceptor to the bipolar cells to the ganglion cell, you have much better acuity, much better discriminative capacity. If you want to read something or see details of a face, or examine something kind of up close, you foveate, and you need light, right? You put enough light on to activate those tightly packed cones, highly pixelated cones in the foveal region, as opposed to the more dusk, dawn, motion detection, lower light responses of the rod cells in the